We have some people joining. Welcome, welcome. I'm going to go ahead. My name is Charity. I'm going to go ahead and go over some housekeeping. I know there's more people joining in, but we'll just take care of some of the housekeeping first while people are joining. Welcome to our webinar, Getting to Know Our Scientific Community. My name is Charity Bourne, and I will be your host. I have worked in applied research centers and labs. I've been a corporate trainer, and I also taught STEM for 7 years. I taught over 3,000 students during that time, and I wrote and implemented around 70 lesson plans for our new science standards, which are called NGSS, Next Generation Science Standards. I now work for a fabulous company called Elim Bio, and a little bit about our housekeeping for our webinar. This webinar is going to be recorded. It will be posted later on our Elim Bio YouTube channel. I know I've already had a lot of teachers reach out to me, and they're excited to be incorporating these into their lessons next year. So I'm really excited to have your questions, be part of them. And you can ask a question anytime during our panelists explaining what they do and talking about their history and how they got involved in science. Questions are always welcome, and you can ask it even when they're speaking. It won't actually disrupt their train of thought and their conversation, because the questions come directly to me, and then I would relay them to our panelists when it's the breaking points. And we'll have a couple times where we'll stop and go over some of your questions that you have for our wonderful panelists. And I wanted to tell you a little bit more about the webinar and why we decided to do the webinar. So today's webinar is sponsored by my company, Elim Bio. And Elim Bio is busy supporting researchers on the front lines. We are proud to say we're helping scientists solve the problem of COVID-19. During this unusual time, we wanted to reach out and help feed future scientific curiosity. We felt the best way to do this is to give you at-home students a chance to meet some really cool scientists in our community. Today's panelist is Stephen Sakuma. Stephen is a science advocate, a biologist, and a PhD graduate student at Sanford, Burnham, Prebys Medical Discovery Institute. Stephen enjoys bringing his work to high-impact, peer-reviewed scientific journals, and to date, his research has been part of 13 published documents. And now, what we've been waiting for, I'm going to go ahead and hand you guys over to Stephen. There you go. Hi.
Hello. Thank you very much for the introduction, Kirsch.
Wonderful, thank you. Well, could you tell us a little bit more about you and your research and what you've been up to with these past couple months?
Yeah, it's been a weird and different and interesting time. I'd be happy to tell you about it. Yeah, let me know if there's too much background noise. Someone just started leaf blowing outside my house. So.
Oh yeah, no, we can't hear it at all. You're good.
Okay, good. Yeah, so I guess I could share my screen. Actually, I have 3 fun facts because I feel like you'd ask
love it.
about.
Yes, yeah.
Yeah. Okay, so yeah, I think one like, I don't know, it's not so fun, it's just more interesting. So I'm the first in my family to pursue a graduate degree. So for me, it's a PhD, but even like at the master's or bachelor's level, or other master's levels.
Yeah.
And then beyond that, I'm the first in my family to receive my bachelor's degree. So it's been a novel journey for me. And then two other fun things. More fun is that I'm an avid snowboarder. And when I was in high school, I raced in the California-Nevada interstate competition all 4 years. And that also, when I was in high school, I was voted prom king my junior year.
Oh, wow. That's cool. So you can be athlete, scientist, and well-liked. See that, friends? Yeah, scientists aren't just in the lab all the time. They actually are real people.
Well, nowadays, sometimes I spend a lot of time in lab, but actually, yeah, I do enjoy my time outside.
Love it.
All right, so we'll share my screen here. All right. Can you see my screen okay?
Yes, definitely. That looks great.
Yeah, so to make this like really understandable, I've kind of reduced this down to just a simple concept. So what I've described as unexpected proteins is more about a strategic approach to looking at ways to treat diseases, which is a little bit different than I think everyone or many people use in the modern day. And I'll explain a little bit more about that in a second. So first question is, so I'm a graduate student of biomedical sciences, and then often people say, like, what are biomedical sciences? Even I, when I was applying, because I originally was a neuroscience undergraduate student, and I was like, what's this biomedical sciences school like? And I think the simple way to think about it is, when you think about biology, you have, the things that make up the living organisms in the universe that go from plants and algae and bacteria all the way to humans. But specifically biomedical sciences are trying to discover things that can then be used for medical application. So this would not necessarily mean making a drug, but it does mean learning something that you could then use to treat a disease. So in this diagram here, you see, switch this to the, you can see like the progression here. So all things in this universe, most normal things in this universe, as they are made up of molecules, including cells, which we'll talk a little bit more about later. But cells are roughly organized into tissues, things you know of, like your skin is on tissue level. Actually, although technically your skin is considered a large organ, for large metric things. And then everything in your body is organized into organ systems. And for us, like, we're thinking not necessarily, and then those organ systems make up the organism, you as a person or anybody else. And we're thinking not necessarily at these levels, but more at these levels. How can cells be changed? How are they altered to change a tissue? And how is that going to affect the function of an organ? So yeah, there are many different types of cells in the body, and they make up many different types of tissues and subsequently organs and systems of the body. I'm not going to go through all this. It doesn't really matter. I'm going to focus here on like a skeletal muscle. Most people know we have muscles, right? Your muscles are important. This is a classic like muscular system diagram. If you look at a muscle, or your arm, it has lots of different muscles that make it up. Inside the muscle are these bundles, skeletal muscle tissue. And each one of those is made up of cells. These cells are actually really interesting because, unlike most cells in the body, they have many nuclei, tens to hundreds of nuclei. But what is a cell, kind of taking things back a little bit? So this is a really cool picture. It's just kind of a stock background for our institute, but I really like it. This is a picture of a cell at very high magnification. And you can see this green dot in the center is the nucleus. And then these kind of yellow things on the outside are proteins that allow a cell to kind of spread out a little bit. And this is like a very common way we look at cells. And I have my work, part of what I love about my work is I spend a lot of time at microscopes looking at and acquiring images just like this. And I often think, oh, wow, this is so cool. And I love taking like beautiful pictures. And I'm like, this is awesome stuff. And yeah, I didn't take these pictures, but I've taken lots of pictures like this, and it's really a fun thing to do. But you can't really see a lot in a picture of a cell like this, so there's a lot of things you can do to look at it in more detail. And through lots of previous research that we are building on, you can see here like kind of a basic organization of a cell. There's lots of stuff here, and then we can discuss it lots if you guys want. But the main thing that I work on is a set of proteins which lie not just in the nucleus, but actually at the edge of the nucleus. So the nucleus is the big organelle in the center of a cell that holds the DNA, which I think most people know is the content of the genetic material that kind of determines how it's the blueprint for the human body, as they say. So when you look at the nucleus, you'll see these complexes here, which we call the nuclear pore complex, or NPC. So we're studying proteins that make up this structure. So the details aren't really that important, but there are a bunch of different proteins, 32 different ones, and we call them nucleoporins. And what's interesting, these are the unexpected proteins, which I kind of described in the beginning. So we're not necessarily looking at a disease, but we're looking at these structures. And then we say, how can changing the components of these structures, how does a little shift in the proteins in it change the way a cell might function, and that would change how an organ would function, and actually affect how the body of a human might function as well. So yeah, and so like people say, oh yeah, this is a really nice like cartoon, but what do these things actually look like? So this is an electron micrograph. These are very, very small structures, and you can't really see them actually with your light, with your eyes, because the size of them is beyond what's called the diffraction limit of light. So they are so small, the size within it, that the wavelength of light isn't big enough to actually reflect back in your eye and give you any sort of meaningful information. So this is an electron micrograph, so those electrons have a much smaller wavelength, so you can see much smaller detail. And the only thing to see here is that you have these very just kind of regimented structure. On the nuclear side, it looks like there's like a basket, and then on the cytoplasmic side, there's like it's like an open basket. And yeah, and so for us, we're basically looking at how these structures do things differently within a cell, and then how does that affect how a cell might function. And so these things, border the nucleus, and this is the cytoplasmic side of the nucleus, and this is the nuclear side of the nucleus. And you can imagine that this is an open pore, and things can go in and out. So this is called nucleocytoplasmic transport, which is just a fancy term for moving things in and out of the nucleus. And this process is well studied, but we and others have found that there's a lot of other things going on with this structure. Lots of things with DNA, which is your labeled chromatin. Yeah, I won't go into the details here, but like it's very interesting how this structure can play such a big role in that other stuff. And then, yeah, so this is just one example of what our basic research would look like. So in this figure, this is a big muscle cell, as we had discussed before. Muscle cells have many nuclei within them. This is one single cell, and you can see that it has many nuclei. So these blue dots here are labeled the nuclei, and the green stain in this image is one of the nucleoporins that I discussed before that make up this structure called NUP210. And you can see it's in green in these muscle cells, but then actually you notice if you look at these cells that are not in the muscle cells are labeled by red here, that they are not they're not green. And what's really interesting is that is not true of all the nucleoporins. So here's POM121. It's labeled in green in this image, and you can see that it is present on these cells. So this is one of the first times that people demonstrated that like a single protein change in this structure can affect, be different between different cells. So two cells, very similar. This one, actually, these ones are muscle cells are derived from these cells, which are precursor cells. So they can begin to turn on or turn off expression of one protein. And so the core of our work has basically been describing how different cells have different composition of these. So in this very simple diagram, we have a gray structure here, which is, you could think of it as just the plain default mode. And then you could say cell B, which has these red things added to them. And these red proteins can change how the cell functions. So I think that's all I had on, the specifics of my work. It's a good time to take a break and take questions.
Yeah, sounds good. We had a couple people that were raising their hands, and I wanted to just make sure that you see at the very bottom, if you have a question, there's a box that says Q&A. So you can type your question into the Q&A, and then I will be able to see your question that you have. So if you had a specific question and you're raising your hand, make sure that you just type in your question into the Q&A, and I will make sure that that question gets passed on to our panelists. I had a couple questions that I had seen come across. And one of them was, what kind of microscopes are you using to be able to see the things? And do you have a picture of what they look like, or?
Yeah, it's really unfortunate actually. I was at my lab today and I was like, I should really take a picture because I don't have any pictures of the one I normally use. It was really sad because I use it a lot. Admittedly, like I use it in the dark, so usually I'm not really looking at the the microscope itself with light.
Well, that's pretty fun for the kids to know that you're using it in the dark. Could you tell them more why you're using your microscope
Yeah,
in the dark?
that's a great question. Yeah, so the picture I showed before was of electron microscope, which I've never used. It's a much more, a very different technique. What I use is a type of light microscopy, which means you're basically shining a light at your sample and then you are looking at or taking pictures of the light that is absorbed and reflected or transmitted, some way you're looking at light. So that's a light microscope. So the reason why I often work in the dark is because light, the light that you're trying to look at will be obscured by the light in the room. So like this room is very bright that I'm standing in, you can see me. But if you can see me, if I'm looking at a microscope, then the light that is reflecting off me now is also reflecting off my sample. So this is a common problem. So it makes it kind of like, it's sometimes fun, it's sometimes a little bit dreary. You're like in this room, it's like, oh, it's really dark. Like I just need some sunlight. Sometimes I walk outside and I'm like, oh, it's so bright out here. But in general, like, yeah, it's not so long, and I'm usually like it's very nice to be able to work kind of in quiet in the dark. It's very peaceful usually. But yeah, so I use a type of microscope, just a little bit more detailed, called a confocal microscope. So this is a kind of an advanced technique which allows you to see like a very thin a viewpoint of what you're looking at, which is really nice because it basically makes it more clear and gives you better resolution, which allows you to see structures in more detail. So those pictures, I think, yeah, here, so these were acquired with a confocal microscope. So an easy way you can tell is that a confocal microscope will give you like a single, very thin plane. So you see this is the center of a sphere. So if you cut a sphere in the center, you'll see the edge be very bright. And while if you do epifluorescence or a non-confocal microscope, you'll see it looks more blurry around the outside.
Cool. And another question was, are the things you're looking at really those colors, or did you color them?
Yeah, so that's a great question. Yeah, so the answer is yes and no. We use pseudo coloring often, so these images are acquired through it's like a gray value. So this image, as it's in its raw format, is just white. So it'd be like a black on white or white on black background. That being said, these colors these dyes that we use, sometimes called fluorophores, have an inherent wavelength that they emit and absorb at. This means I provide a certain light, and then they absorb it, and then they emit a certain light, and that light is actually green sometimes, it is blue sometimes, and it is actually red. What's really cool about using a camera as opposed to your own eye to just look at the images is you can look at things beyond your own ability to see. So the visible spectrum for a human, goes from a very far violet to red, a dark red, but we can actually acquire images in the infrared. And so obviously that color has no real meaning in human discussion because I can't see infrared. What does infrared look like? I don't know, it looks like red most of the time. But that being said, yeah, many of the colors actually look this way. So when I look in the microscope, I do see things like this.
Wonderful. And then you mentioned that the proteins are unexpected. What makes them unexpected?
Yeah, it's a great question. I think the core thing here is going back to what I had originally said about like approaches. So it was unexpected in the sense that people generally were thinking of like, so this is muscle and this is, had some possible applications for treating muscle disease. So people are concerned in muscle about like how muscles contract. So there's a very well studied like kind of contractile apparatus. There are proteins that like kind of move along fibers in the cell, and that brings the cell to like suck it in. And that has no direct bearing. The nucleus has no direct bearing on how that works. And it was unexpected in the sense that no one had thought, oh, what is it that what is it that the nucleus might be doing, these, specifically these set of proteins, no one thought, what are these proteins doing that might be important for that process?
Wonderful. And then another question that we had just pushed in is your work basically about restructuring certain proteins in order to change the response? Or is that a clear understanding of what you're doing, or?
Yeah, so I would say the answer is yes and no. I feel like in science, oftentimes the answer is yes and no, because things are just very complicated. And in the short answer, like, so in theory, so from a basic level, it's not necessarily that we are trying to restructure the cell. We're trying to understand why the cell restructures itself. So I'm not asking, oh, like, if I change this, like, is that important for like the cell's function? That is a test question. That is how I would test my hypothesis that this thing is important for that process. But in the end, we may actually end up using that. So we may actually change. That's actually what happened in this paper you see here. It's not in this figure, but we changed the composition by blocking the expression of NUP210, the green protein on the left, and we'd see that it affected the cell's ability to turn into a muscle cell. And although that is not necessarily the way this would be utilized for a treatment for a disease, it is a way it could be. So you can imagine, oh, if you don't have good muscles, maybe you should increase expression of this. So that was a hypothesis that was generated directly from this work, and I think it still hasn't been shown whether or not that will actually work. But even if that ends up not being the way to approach it, you learn a lot from this that you could then utilize in another way.
That's a good lesson about science in general. Sometimes you like you think you're going down this path, and all of a sudden you're over here.
Right. Absolutely.
The follow-up to that question from the same person is, does this response ever change how your cell affects to foreign antigens or pathogens or viruses to the protein?
Yeah, it's great that you asked that question. I mean, obviously, I think people are very concerned about foreign pathogens related to COVID and SARS-CoV-2. And so the answer is yes. And so it was a very unexpected finding and very cool, which we published, I think, 2 years ago now, that it actually, this exact same protein, so we had done a lot of work with these guys in cell culture, and we had found a way to explore the function of this in mice. And we had found that mice T cells, a type of immune cell, its function is changed by function and survival is changed by the absence of this protein. So we found that actually that this same protein, which plays a role in muscle function, plays a role in immune function, and how cells of the immune system could respond to pathogens from the outside.
Wonderful.
Yeah, and another example of unexpected things in science coming about. But this actually, I think, underlies the thing I was saying originally, which is just what makes this unexpected was just that it wasn't thought that there was really a connection. I mean, it's still not sure what the connection between the muscle and the immune aspect of what I'm describing here is, but it wasn't thought that there was any connection between the structure and these two very different things. And if you were focused very much on studying the immune function of a cell, this is not the first place you would look. So this is how exploring something from this perspective can give you a lot of insights into that other information.
Cool. Well, you mentioned something about your publishings, and we had a question from someone on the call. They were wondering if all of their things you published have all been about this same topic, or are they different things that have to do with science? They were just wondering more about that process.
Very, very, very varied things, but all basic science. So I have done some other work, but not associated. So when we talk about the publication that she was referring to earlier, we're all peer-reviewed publications. This is a method in science to verify that the results are at least comprehensible from a scientific perspective. So all of my work from that perspective has been in things that are like basic biology. I'd say about half or something like half of it was on this topic, on nuclear porins and nuclear pore complexes. And then the other half was in a variety of other perspectives, because I used to work in other labs, which I can tell you a little bit about. I have some slides talking about it later. But in those labs, we explored various other things like metabolism and lung function, and diseases like cystic fibrosis or pulmonary fibrosis, I think. And yeah, so various things, but all basic science.
That's great. And it's neat for people to hear that you can be focused in science, but also have different things that you study in your path along the way.
Yeah, and most scientists do. I think a very common path is you will start, and you start like kind of in a field like, oh, I'm going to study immunology because I've heard a lot of people like they've changed their kind of their trajectory of their career because they really want to study these infectious diseases that are affecting us so much right now. And then maybe they like are working in immunology and they realize that, SARS-CoV-2 specific studies are very, very saturated, and that actually they're really interested in studying the next pathogen that's going to be a problem for us. So they really shift their things to novel discovery of diseases. That's a very common thing. You take like this topic and you'll say, oh, I'm gonna focus a little bit more over here, and then, oh, maybe I'll focus a little bit more right here.
Wonderful. One more question about proteins. Someone said you mentioned that the structure or the shape, are they all the same shape or structure, or do they change?
Yeah, so it's a great question, and so there are proteins have what's called different conformations, but it's usually a limited number of conformations. They don't have limitless numbers. So you can see it a little bit here. So these different structures are made up of very specific shapes. So obviously, no random protein could make up that shape. Oh yeah, sorry, I don't know if the screen was showing. No random structure can make up this shape, and very specific proteins make up these shapes. And actually, this structure is really cool. Like most large protein structures in the cell, they all have to have like interlocking parts. So it's not just that like, oh, I've got this protein and this protein and this protein. They actually kind of like lock together. So actually, this that here, the base is a ring, and this ring actually is made of many proteins that are like locked together in a concentric circle. So they just kind of fit together. So the shape of protein is very key in that, the placement of one or the other is not arbitrary. It actually matters a lot. And so like the proteins that we were studying in this paper, like NUP210 actually lie right here in the middle part, and POM121 and NUP210, they both can kind of play a similar role. They're in a very similar place and they have a similar shape, at least from a general perspective. There are a lot of ways in which they are not similar. But at least they both can kind of be in the same place at different mutually exclusive. So one could be there, or the other one could be there, or they both could be there.
Cool, wonderful. Would you mind sharing us a little bit more? I think you had some more slides that you wanted.
Yeah. So the next slide was actually just talking a little bit about the space I worked. So this is a little bit messy looking. It's a very raw look. I didn't doctor this photo for you guys to make it look more pretty than it is. So this is my old desk. We actually moved our lab here. So this is where I'd normally sit, and I work on my computer here. And this is my lab bench. And then, so this is a very common setup. Usually you'll have basically a desk next to a bench, and then maybe another desk over there, and then with a bench next to it. But I would spend a large portion of my day, probably half my workday, oscillating between these two positions. I'd be in this chair, and then I would get up and sit in this chair or walk over here and go through these things. So I like to describe this actually because with the work from home and quarantine order, I've realized one thing that I really enjoy about being a scientist, bench scientist, is that it's a very like active job, but in like a very like kind of pleasant way. It's not like a hard labor where like I'm outside like sweating all day long and I come home and like my muscles are sore, although I wouldn't mind that so much. But it is like, it's not so sedentary. So like my boss who is a much more senior scientist, he spends his whole day writing and reading, and he spends a lot of time at his computer. But for me, working at the bench, I spend a lot of time, like I do spend some time at the computer. I have to analyze data and stuff like that, but I get up and I walk to my bench, and I type out things, I can stand, and I can move around. So like I have a pedometer that tracks my steps, and when I'm working in the lab, I walk around a lot, and it's really nice. But yeah, so this is kind of a basic view of what that looks like. I just can't really see a lot of the tools we use. This is a centrifuge. This is a very basic biology lab setup. We don't have any like specialized equipment because we don't work on anything too rare, let's say. Another common place I spend a lot of time, so maybe another 25% of my time, is in a cell culture room. So this is just a stock photo. This isn't from my lab. So this is a very simple microscope here. This is what would be like just like a viewing microscope. So this is like a dish of cells. This is what cell culture looks like. These are called biosafety cabinets, and people work with them. We don't work with hairnets, but everything else is pretty similar. This is a lab coat, so you have what's called PPE, or personal protective equipment. You put it on over the top to protect your clothes and yourself from anything that might spill on them. And so you can look at cells that have been growing in dishes that you plate there in a microscope like this, and then you do the work in a hood like this because these cells are growing in media, which is like a solution that is very well designed for growth, and that bacteria or fungus would grow in if you gave it the chance. Because I'm not studying bacteria or fungus, I'm studying mammalian cells. I don't want those things to get in there. And so that's a little bit about the space I work in. I don't know if anyone has any more questions about that. That's the last slide I got on that part.
I didn't see any questions about the space you work in. It was really cool to see the pictures. And I love what you said about being able to move. I really appreciate being able to move, and I realized that being home too. There was another question about about protein, and it is: do you think that a missing protein in a cell could cause an accidental complement response?
Yeah, kind of yes and kind of no. So I know, so complement usually refers to like a set of a cascading pathway that's allows for clotting in if you have like a scratch or something, and various other pathways in the body. And so a missing protein usually wouldn't cause something like that because it's not really the type of thing that is designed to be activated in that pathway. But there is a way in which it could, and this is where like the classic biology is just incredibly complicated comes into play, which is that a missing protein can cause a lot of weird things to happen in a cell, all the way from just like a minor malfunction, like a 10% reduction in its speed or its ability to do something, or it could cause something very drastic, like the cell to die or stop functioning altogether. And if a cell dies, it can cause a lot of very abnormal things to happen. It can cause this, a clot to form, or abnormal like aggregation, or a bunch of like inflammatory molecules in your body. So yeah, so there's a lot of ways that could cascade into that, but not in general, not specifically, because that's not the way that pathway is specifically designed. But it's possible for sure.
Great, thank you very much. And I noticed that when you're in your lab, there was a lot of people in the bench next to you. So do you get to work with other people in your lab, or are you working by yourself usually?
Yeah, so that's a great question. So this room picture here is somewhat similar to the room that I work in now. We have there are 4 hoods in this room. The arrangement is totally different in our room, but we also have 4 biosafety, or we will have 4 biosafety cabinets, and one like this. And so yeah, so I think this is a very not a abnormal setup here. Like I had my previous lab, we had actually 3 in a row, and we would often have 3 of us sitting right next to each other. We would all be working, and we'd be talking to the person next to us, and it was kind of fun because one of the cool things about bench science is that it's a very there are a lot of things where you have to like focus a lot, and you have to think about and calculate like what you're doing. But a lot of the times it's like, oh, you calculate a number, and then you use that number. So like, I need 5 milliliters of something. I need to pipette it 50 times. And you're pipetting, and you don't require a lot of thinking. You're just moving your fingers or passing things around. So you can talk to people next to you, ask them how it's going, but also you can ask them about work. You can be like, oh yeah, I'm actually trying to do this, and they say, oh, actually I know a better way to do that. And those are the greatest, because then you feel like you really have colleagues you can appreciate. And yeah, and so the same thing is true in the general lab space. Here though, it's a little bit more complicated because people are working at computers, so sometimes there's an expectation maybe not to be talking too much so that people can focus if they're working on like a computer task, so they have to type or write. But usually the precedence in this space is for lab work. So if I'm working here and my colleague is working here and we need to share something, nowadays we don't work that closely because of social distancing guidelines. But we're just like, okay, yeah, turn around, I can just pass you something or tell you about how to do something better or worse. And yeah, so this is like kind of the small lab environment, but we also have external collaborators, other labs who you'll have meetings with every once in a while to discuss the progress of your projects and how the science is unfolding. Yeah, so those are the people you commonly work with. And then we also work with a host of other people who are just in what's called like core facilities. There are people who specialize in a specific task, working with this type of microscope or that type of machine. And those people you all associate with to try to get your work done.
Wonderful. And when you're collaborating and talking with other people, do you all share information equally? And
yeah. So I mean, generally, academic science is pretty open. It's famous for this. So people who work in a company, maybe they're what they're working on is very top secret. It's like I'm not going to tell you because then you could sell it to this company and then they would steal the idea from us. In academia, it's much more open. It's not perfectly open because there's always the worry that you publish a peer-reviewed work. A lot of the publication itself, a lot of the publication process is verifying that what you're doing is novel. It's new. No one has done it previously. So if I tell someone who's a direct competitor and I say, oh yeah, I'm working on this thing, then they do that and they publish it before me. It's a problem. But it's very rare. 99% of the time, the people I talk to, they would never be interested in working on the thing I'm working on and would never be able to do the things, faster than I'm already doing them. So yeah, like I'll talk to, when we're sitting at lunch, actually, oftentimes we'll be sitting there and just be like, oh yeah, we're working on this assay to do this thing. And someone's like, oh, wow, that's really cool. And actually, scientists are the worst for talking about work. I think people always say, oh, someone's talking shop, like, oh, you're talking about your customers and you work at like a store or something. For us, it's the absolute worst. You have a bunch of scientists, they're in like a proper like happy hour or party or something, and they're like, so what do you think the role of this protein is in this context? And everyone's like, oh, nerd. But the problem is that science, and this is what I think what makes this happen, is that people are just so passionate and they're so engaged, and it's so interesting. Like sometimes I'm not trying to do science. I'm like just like surfing the web, and I like find something, and I'm like, that is so cool, and I'm just like, I have to know more about it.
I love it. Well, what do you like to do? You mentioned surfing the web. What else do you like to do when you're not working?
Yeah, so actually I'll continue down my presentation. I just have one more thing because I think it's a good segue to that. So it's a little bit about my background. So a little bit about me. So this is a picture of a mountain. This is Mount Lassen. I grew up in a very, very small town in Northern California. It has less than 1,000 people, or maybe more than 1,000 people. But it generally looks a lot like this. This is, and I think when people talk about small towns, I remember when I first went to college, I talked to people and they were like, oh yeah, from a small town, like it's outside of LA. And I was like, I don't think there are any small towns outside of LA. Like there are small towns relative to LA, but that's not a small town. This is 1,000 people, but it's not just small, it's also very rural. It's in the middle of nowhere. So like, this is not just the way the mountain looks, this is actually the way the town looks because it is 1,000 people, which means there could be 1,000 people on the other side of these trees. It doesn't take a lot of space to hold 1,000 people. So yeah, it's a very rural area, and these I think people often ask about like difficulties, and I think coming from an area that doesn't have a lot of like urban infrastructure, is like a huge kind of difficulty because, you know, I did not know what a scientist was. I think it's really great that this webinar exists, by the way. Thank you guys very much for putting it on. This, like, when I was a kid, I actually did want to be a scientist when I was in the 5th grade. And I thought, wow, this is really cool. I remember we did a dissection of a salmon. We went to a fish hatchery. We got a salmon and we got to like look at all the parts of the salmon. And I was like, this is cool. But I still didn't know what a scientist was. But I said I wanted to be a scientist for a few years. And then I decided I wanted to be a lawyer for some unknown reason. And I totally forgot about being a scientist till I got into college. But speaking of which, I went to college at UC Berkeley. So it's one of the most prestigious universities in California. It's also in Northern California, but well south of where I live. So it was a bit of a trek and quite a bit of a move away from my home. And when I was there, I became really interested in science. Like everyone I knew was into medicine and pre-med, and all the pre-med people do biology. They're like, oh, you should try taking this biology course. And I took it and I was like, wow, this is really hard. And it really was not my strength. Like I was exceptionally good at math and physics and even writing. Actually, my biggest strength was probably in the humanities, but I loved the biology. I was like, this stuff is so cool. And I was like, I don't want to do medicine. Like dealing with like people. I love like contributing to society in a good way. But interacting individually with people, I was like, that's not the way that I want to contribute. Like I want to contribute to like a big picture. It's not like I cured your disease. I didn't give you a drug to help you out. I gave society a drug, society information. I was like, and I'm a bit of a purist. When I started my graduate program, I was like, people ask like, well, what do you think about this process? Like I think about contributing to the vast body of knowledge we think of as science. So after I graduated with a degree in biology, I went to work at UC San Francisco, so this is just across the bay from Berkeley, and I worked for a basic lab there looking at lung biology, lung functions, and also metabolism. And we can talk more about that if you guys are interested. And then I moved to Sanford, Burnham, Prebys Medical Discovery Institute, which is a bit of a mouthful, and I worked there for a little while before I started my graduate training to get my PhD. And I think, yeah, so that's it. And then I have this question I like to ask in general, but we can get to it later.
Well, I just wanted to share that you had one of the attendees that just wanted to make sure you knew that you're very lively and fun, and they think,
oh, awesome.
They think it's fun that you do other stuff like snowboarding and other things.
Yeah, and so I'm happy to tell you a little bit about that too. So yeah, I love snowboarding, but I live in San Diego now, and it's quite far to the nearest mountain, and I still go at least once or twice a year, but not nearly as much as I used to. When I was a kid, like I grew up very close to a mountain. I would go like, I don't know, like 50 times a year or something, like a ton. And yeah, so, but now I tried to pick up surfing, and I found that, it is super fun, but it's not my favorite. I did triathlons for a while, and when I was surfing, I think the problem is I'm a very active person, and surfing is a lot of like calculating and waiting and planning. And I'm like, I just want to like use my energy. So like sometimes I'd be surfing and I would just be like, this isn't a good wave, but I'm going to paddle anyway. And then I was like, maybe I should just swim more. So I like to swim. And in triathlon, you swim, you run, and you bike. And I do a lot of those things in general. Like I love swimming in the ocean. I actually bike to work, and I bike just for fun around San Diego. And I run for exercise in general.
Wonderful. Do you ever find when you're switching between, when you're doing a triathlon, that it's hard to switch between the different running?
Oh yeah, absolutely. And especially if you've been training in one for a super long time. Like I haven't been swimming a lot recently, and I go and I'm like, oh my gosh, the swimming's so hard. Like,
super fun. Well, what was your question about passion? Did you have a question?
Oh yeah, so my final point I think would always be to bring around, so like, I am obviously a biologist and a scientist, but I think a lot of things I always try to convey to people when this perspective, I think this is the perspective of a scientist, and I think I can apply this to my own life in retrospect. Like, when actually I started at Berkeley, I was good at math and physics, and I thought maybe I should go in those type of directions, and I really thought actually maybe I should be an engineer. And I thought, I really think even now that that would have been a really great path for me. And the main reason that I didn't do it was because I felt very strongly that I, um, I needed to like finish, because engineering is a very challenging degree like biology. And I had already kind of started down the path of biology, and the switching would have been difficult. It wouldn't have been impossible in any way. And I think it's okay to follow that passion. And I'm happy that I did what I did, and I think it was okay. But for every student out there, and you're trying to think about what you want to do with your life, like ask yourself, like, what is your passion? Like, you don't have to be really good at something. You do have to be like passively good. Like, if you are not good at something, you want it to be your passion, just don't make it your work, because you don't want to be like not good at it. That being said, though, practice goes a long way. Like, biology was not my strong suit when I first started, but now I'm very good at it. And I think that you don't want to give up too early. And if you really do enjoy something, just be like, and maybe there's some barrier, maybe too, like maybe biology is your passion, but you really don't like this aspect. Like, actually, didactic is what you usually call like classroom learning for biology. I find particularly troublesome, annoying, and I think it's a very important perspective to have, because you have to remember, and I think that's probably one of the big things I would recommend, is that what you learn in school is not the job associated with the work. So when you are learning to become a biologist, you're not doing biology. You're learning about other people doing biology. And when you do biology, it's not going to be anything like that. Like, biologists don't sit down with a book reading about, things and then you're writing them out in tests. They do things physical, and that applies to almost all professions.
Totally. I just had a question actually about science as your profession. Do you think of it more when you go to work more of as a job, something you're going to, or do you think of it as a passion and something that energizes you?
Yeah, so I would say it's a combination. Definitely the passion component is there, and it's what drives, I think, the mind to work well. And then the thinking of it as a job, it drives the mind to work consistently. Because I think it's easy to be passionate when you're passionate and you're interested, but it's hard to be productive if you are just writing your passion. Like, I am driven by the need to, for the sake of my degree and my boss's expectation of my job, to continue doing things. And I think it's actually a kind of a valuable lesson in life. Like, you can't always just do like the thing that you want to passionately. Like, I have things I want to study biologically that I can't, not because it's impossible or anything, but because we have a focus. We have some expectations. Like, I need to finish things. And that is something I think in life in general. Like, oh, you may want to like buy a house or a car, but if halfway through the paperwork you're like, I'm tired of this paperwork, like, then you can't do it. Like, it requires you to finish the steps to get the things you want.
Yeah, a lot of life is steps. I agree. Like, you have to do one step and then the next step. And in each level that you're at, you might have moments where you're like, yes, and other times where you're like, oh, okay, here I go again.
Yeah, exactly. And I think, yeah, a lot of times in my like my undergraduate career and even now, like, things are very tedious. Like, I took organic chemistry. A lot of people hate organic chemistry. I loved organic chemistry, but there were times, even in the subject that I loved, where I was like, oh my God, the nomenclature and the shapes, and I'm like, what is this thing? And with my work, like, we have, very repetitious work. Sometimes we have plates that are about this big, and they have 384 wells in them. They're tiny, and you have to pipe at one thing in every well, or two things maybe in every well. So that's 600, 700, more than 700 pipetting, like, and it gets tedious at times. But what the quality, what you can get out of that is like, it's like magic. I think it came up in Breaking Bad. I still remember the first time I heard it. Like, the main character, he's like describing science, or someone talking about science, and he's like, it's like magic, and it really is sometimes.
It is. Yeah, that's what I used to teach. When I would teach kindergarten science, they always thought I was doing magic. Did you have any changes to your work schedule with everything with COVID-19 or is?
Yeah, so huge changes, obviously, I think like most people. So we had a general shutdown and only essential work was necessary, allowed to be done. Um, and so that basically meant just maintenance tasks, making sure that, you know, things that we had living kept living and didn't die in ways that we didn't want them to. Um, and I spent a lot of time working from home, so that's where I am now and where we're expected to be whenever we're doing things like this on the computer. But now I go into the lab kind of in like kind of precision strikes. I have to like, you know, tell my boss, like I'm going to do this, this, and this. I'm going to go in from this time to this time, and then I'm going to be gone. And I'm going to be home, you know, analyzing data or whatever it is. So now I'm spending, I'll probably switching now. We're just trying to transitioning back. I'll probably be at least 75% of my days in general per week will be in the lab, like doing the pipetting, the stuff that you have to be in lab to do. And the rest of the time I'll be at my computer at home analyzing data.
Some days you're in the lab, some days you're not. Do you guys have to rotate who's in? Do you have a schedule?
Yeah, so most people have rotations. Our lab is kind of a little bit small for the space that we have, so we're very spaced out, so we don't have to worry. Like, I don't, even if we're all in, we don't actually have to be very close. I can work 10 feet away from the next person, and we all, like 6 of us, can be in at the same time because we have 60 feet. It's just math. But yeah, for a while there, we had schedules, and we were basically just either the morning or afternoon. But yeah, right now we're kind of opened up from that.
Yeah.
Although that being said, actually going back so about the cell culture room, so the space is a little bit smaller there, so actually only one person can work in each space at a time, which is annoying because, you know, you pay a bunch of money for the setup for both of these things to be usable, and now only one of them can be used at a time.
Yeah. What about your supplies that you're using? Do you have to clean them differently? I know that in the schools, they're going to have to start sanitizing things differently than they have before, or is it you were already doing that anyway?
Yeah, so one beautiful thing about biology is that, and science in general, is that it's generally done very cleanly to start with. So most of the basic work I have has no enhanced cleaning process. So I wear personal protective equipment, you know, goggles and gloves and a lab coat all the time. So I don't touch anything and with my bare hands, and I use my gloves, and I don't touch my gloves to my face or anything. So there's no increased exposure there. The place where we work, the handles and the doors and all that stuff is being sanitized by the cleaning staff more often, and we are just generally advised to like, be cautious and not just be careful and wash our hands more often. I think this is a generally good thing to do.
Good, wash our hands. Definitely. So we have one more question. It's kind of a challenge question for you. You ready?
I'm ready. I love good questions.
So we have a kindergartner on the call, and they think of a molecule like a balls. Can you explain it in a way that would maybe make a little bit more sense to them than just balls?
Yeah. I think the ball analogy for a molecule or an atom or whatever is great, but it breaks down because we have a limited view of the number of balls that exist in the world. Like we have a basketball or a baseball, but the number of molecules that exist is incredible. There are millions, more than millions, yeah, probably easily billions of different types of molecules. Maybe the number is limitless, who knows? But you can think of it kind of abstractly in a lot of different ways. So it's not just a ball. It could be a stick. It could be molecules can take basically any shape you want. Like some people who do molecular like engineering, they design molecules that have just kind of fun shapes. They have circles, and you can write out words with molecules. I remember I saw a really cool picture one time. Someone had written, what did they write? I can't remember, but it was some sort of word. It was like hi with molecules. So, I mean, molecules as the building blocks of everything we see around us, they are the building blocks of everything we can use to imagine the things around us. So if you're thinking, oh, is a molecule a ball? Yeah, molecules can be balls. They can be sticks. They can be disks. They can be sphere. I guess sphere is a ball. They could be like plates, like a sheet, like a piece of paper. But I think a good way to think about it is if you think of atoms, atoms are kind of more like, it's still just an analogy. Atoms are what makes up molecules, and they are just balls. But you know, how you organize those balls is how you get a certain type of shape. You got a ball right here and a ball right there, and you have a line of balls, and then you have a stick.
Yeah. My kids have this one toy, and they're little magnetic balls, and you can put them together to build different things. Have you seen that toy?
Yeah, like connects or not.
Yeah, but these are magnetic. But yeah,
Oh yeah, you're right. The magnetic ones, that's different. Yeah,
yeah.
I guess I haven't seen that.
So maybe that would be a good thought process for the kindergartner too, is that they can be built into different shapes and
Yeah.
Yeah.
Yeah, actually, a pretty good analogy. Actually, now that you mention it, actually, I'm thinking even more abstractly. A good analogy might be like Play-Doh or something malleable. So Play-Doh can take any shape. It is this kind of substance, and you could roll it into a ball, and you can make a little ball. You can you could hang it upside down. You can make a bracelet. There are a lot of things you can do with Play-Doh, and it is shapeable. But it has kind of an inherent quality, and that also kind of limits its abilities, right? So like Play-Doh, you can make a piece of Play-Doh and you can make it really long, but there's a maximum length, right? You can't make, maybe you could make a mile-long piece of Play-Doh, but you probably couldn't lift it up, right? It would be too much force and it would be pulled apart. So that's kind of a way of understanding like the limitations also of the material that you're working with, molecules or anything else.
Oh, that's wonderful. I love that. That's a great analogy.
Yeah, I've never thought of that one before. I always try to think of good analogies for really like obscure things from something is small enough, it's so obscure. It's like, how do you even describe it? Like, that's why we think of atoms as balls, because we don't really have any other way of thinking of them or visualizing it.
Yeah. Well, I always think the students have the best questions. They get us thinking as well about how to explain things, to articulate it and bring our thoughts to everyone. So I always appreciate the students for that. They do a good job. And you did a great job too. As the one of the attendees said, you're very lively, and it's been very fun talking to you. We really have appreciated your time and your ability to share what you're going through with COVID-19, and also all your research that you've done. That was super exciting and interesting to find out about. And we really appreciate your time, Stephen, and wanted to thank you for being part of our getting to know our scientific community.
Thank you very much, and thank you everyone for listening in, and hopefully it was very, very useful.
Wonderful. Thanks very much. Have a great day and have a wonderful weekend.
Thank you, you too. Bye bye.